When you look at the back of a multivitamin bottle, there is an expectation of total safety. You see a list of everyday minerals, calcium, zinc, maybe some vitamin C, and right there in the middle is iron. Most of us swallow the pill without a second thought, treating it as a mundane dietary requirement. But in an emergency department, a toddler finding a bottle of concentrated iron supplements is a life-threatening crisis. Within hours, the metal acts as a corrosive, burning the mucosal lining of the stomach and intestines. Doctors monitor for vin rosé urine, a distinct pink-red color in the catheter bag that provides the only proof that a chemical antidote is successfully stripping the poison from the blood. This danger is rooted in the history of the Earth itself. In the primordial, oxygen-free oceans, dissolved iron was abundant and accessible. But when photosynthetic life began flooding the atmosphere with oxygen, that iron oxidized. It literally rusted, precipitating out of the oceans to form these massive, solid layers of red rock. This shift created an evolutionary crisis. The element all life depended on was suddenly insoluble and, in the presence of oxygen, highly toxic to the cells themselves. To survive, multicellular organisms were forced to build heavy internal shielding to transport and store this metal without triggering oxidative death. We still live with this biological paradox. Iron is the essential engine behind every breath you take, but it requires a perfectly calibrated security architecture to prevent the body from being shredded from the inside out. Iron's core chemical superpower is its flexibility. Under biological conditions, it easily passes single electrons back and forth, transitioning between a state with a 2+ charge and one with a 3+ charge. It is a microscopic battery that never stops charging and discharging. This single electron handoff is the specific mechanism that allows hemoglobin to bind onto molecular oxygen in your lungs and then release it in your tissues. It also shuttles electrons inside your mitochondria to generate the energy that keeps you alive. Because of its utility, the body keeps 65% of its iron active inside circulating red blood cells. Another 25% is kept in deep storage within the liver and spleen, acting as a metabolic reserve. Yet the actual vascular transport pool, the iron moving freely through your veins, is kept incredibly small, making up less than 0.1% of the body's total iron. This extreme restriction in the plasma is a direct result of how volatile this fuel is when left unprotected. If free iron bumps into naturally occurring hydrogen peroxide inside a cell, it triggers the Fenton reaction. The iron splits the peroxide apart, unleashing hydroxyl-free radicals that act like molecular chainsaws. They tear lipid membranes apart, cross-link proteins, and cause double-stranded breaks in your DNA. To prevent this, the body uses dual containment architectures. It stores iron in spherical protein cages called ferritin, which forces the metal into a safe mineral core. For transport, every atom is gripped by a specialized escort called transferrin, which ensures the iron cannot react with anything while traveling through the blood. This system is supported by a massive recycling operation. When red blood cells wear out after 120 days, immune cells called macrophages in the spleen and liver swallow them whole. They extract the core iron and immediately hand it back to transferrin to be built into new cells. Because we are so efficient at recycling, we only need to absorb 1 or 2 milligrams of new iron from our diet daily. However, regular blood loss during the menstrual cycle requires adult females to double up daily intake to prevent the system from running dry. While we take in small amounts of iron to replace what we lose through skin and mucosal cells, our survival is anchored by the massive scale of this recycling loop. We function on the precision of a zero-waste economy. In the genetic condition hemochromatosis, this precision collapses. A mutation makes the liver blind to iron levels, so it never signals the gut to stop absorbing. Over decades, the body fills with unshielded iron that spills into the organs, turning the liver and heart into rigid scar tissue. In a healthy system, the liver prevents this using a hormone called hepcidin. When iron levels are sufficient, hepcidin travels to the gut and binds to ferroportin, the exit doors that let iron into the blood. Hepcidin doesn't just lock the door, it triggers its complete destruction, forcing the cell to swallow and digest the channel. This mechanism is also a rapid response weapon. During an infection, the immune system releases hepcidin to lock iron away inside your cells. By removing the exit doors, the body starves invading bacteria of the metal they need to multiply. This immune response creates a diagnostic trap. It causes ferritin levels to spike as the body tries to hide its iron, which can mask a severe underlying deficiency behind a lab result that looks like plenty of storage. To see through this illusion, pathologists check the soluble transferrin receptor. While ferritin levels can be skewed by inflammation, this receptor only increases when the tissues are truly starving for iron, providing a stable indicator that cuts through the noise. Correctly diagnosing an iron disorder requires looking past standard lab numbers to see how the immune system is actively manipulating the metal for defense. In the lab, serum iron is measured using colorimetry. Automated analyzers use an acid buffer to strip iron from its protein carriers and bind it to a chemical ligand that changes color. The depth of that color is directly proportional to the amount of iron in the sample. But the measurement process has a common vulnerability. If a blood draw is performed poorly, mechanical force can burst red blood cells in the tube. This spillage floods the serum with hemoglobin iron, creating a false positive that can lead to a dangerous misdiagnosis. Clinicians evaluate risk using the transferrin saturation calculation. A result exceeding the 50% threshold indicates the armored cars in the bladder fully saturated, leaving the system with no more room to safely capture reactive iron before it spills into the tissues. Timing is also critical due to iron's extreme diurnal variation. Levels naturally peak in the early morning and can drop by 50% by the evening. An iron panel drawn late in the day can easily mimic a deficient state, failing automated laboratory quality checks. This variability makes diagnostic stewardship essential. Ordering repeated, daily iron panels on stable patients is clinically unjustified, and through repeated blood draws can actively contribute to the patient's anemia. Monitoring this metal requires exactly as much discipline and strict boundaries in the lab as the human body uses internally. Inside your gut, a microscopic war is constantly happening over this metal. Trillions of bacteria deploy specialized high-affinity scavengers called siderophores to capture dietary iron and steal it before your own cells can absorb it. When the body is overwhelmed by a sudden, massive dose, as in an accidental overdose, standard treatments fail. Activated charcoal, a common emergency antidote, cannot physically bind to iron ions. Instead, the medical team must use darofoxamine, an intravenous chemical cage. It hunts down free toxic iron in the plasma and binds to it, creating a water-soluble complex that the kidneys can safely filter out of the body. For chronic iron overload, the gold standard treatment is the regular removal of whole blood. It's a modern application of the ancient practice of bloodletting. By removing blood, doctors force the body to drain its toxic iron stores to build new, healthy cells. From the competition with gut microbes to the emergency use of intravenous cages, the biological reality of iron remains the same. It is the essential spark of life when perfectly caged, and a lethal corrosive when free.